Thanks, Miho. Good afternoon, everyone. Thanks for coming. My name is Charles Bai. I'm representing Vector Builder to give this speech about in vitro transcribed messenger RNA from design to therapy, part 3. The focus of today's presentation will be about one-stop solutions for mRNA drug development provided by Vector Builder. To give an overview about today's seminar, we're going to talk about the codon optimization, the quality control, and the Vector Builder's solution for RNA development. To produce in vitro transcribed mRNA, you start with constructing the in vitro transcribed mRNA vector containing certain critical components, such as a T7 promoter to drive the expression, transcription of a 5' UTR or gene of interest, 3' UTR, and the poly tail. Once the cloning is ready, the vector needs to be linearized at the end of poly tail to be used as a template and mixed with the T7 polymerase and the ribonucleotides A, U, C, and G. The reaction will transcribe from the beginning of the 5' UTR all the way to the end of the poly tail. At this stage, there's still a critical component missing on this mRNA, which is a 5' cap. To add the 5' cap, there are basically two popular approaches. The first one is called enzymatic capping, which happens after in vitro transcription reaction. The products will be mixed with two different enzymes to apply the cap1 structure to the final product. And another approach is called co-transcriptional capping. What is different for this method is that in addition to T7 polymerase and ribonucleotides, you also need to add a ribonucleotide analog, which is a trimer of the ribonucleotides, as it is shown in this dashed box. And this trimer has already contained the cap1 structure, and it will be used as an initiation point for in vitro transcription reaction. As you can imagine, every nucleotide being added after this trimer will become the IVT mRNA, and they all have added the cap1 structure. To apply the IVT mRNA in vivo for good translation, using naked mRNA will not work. As we showed here, we inject a different amount of luciferase mRNA into mice, and you see barely no expression at all at 6 hours, 24 or 48 hours post-injection. Once we encapsulate the mRNA into lipid nanoparticles, we see good expression of luciferase 6 hours post-injection. To further boost the expression of the luciferase mRNA, we use a modified nucleotide, which is called IM1 PSI, to substitute all the uridine in vitro transcription. And now we see the best expression of the luciferase mRNA compared to the previous ones. Furthermore, using pseudouridine can also ease the mRNA-triggered pro-inflammation reaction. As you can see here, we collect the serum 48 hours post-injection and quantify two pro-inflammatory cytokine expression. And the mRNA made with the M1 PSI had less expression of those two cytokines compared to the one made with just normal uridine. So looking at the components of mRNA, we know that the choice of UTRs are really critical for the expression of RNA. In this case, we test a different combination of human endogenous UTRs, and we found one of them had a much better expression compared to the rest of them. We also know that the length of a poly tail is also critical because it is basically stabilized RNA, increases the expression time of the mRNA in cell. So the longer poly tail, the longer mRNA can stay and to be translated. In this case, we compared EGFP mRNA with 110 base pairs or 60 base pairs adenine. And you can see the one with longer one expressed much better at 96 hours post-transfection. So looking at the very core of this mRNA, which is the coding sequence for your gene of interest, it can also be improved through codon optimization. And the codon optimization mainly done to achieve three purposes. The first one is the GC content and repetitive sequence optimization, which is to help the vector cloning and vector stability. The second purpose is to choose the preferred codon in the targeted system for the mRNA to be expressed. The third purpose is to optimize the secondary structure of a single-strand mRNA, which is vital for the translation efficiency and mRNA stability. So based on the three principles, we developed our own optimization algorithm. And here is an example when we apply the algorithm to a reporter protein luciferase, firefly luciferase. So the red line here indicates the expression of the firefly luciferase wild-type mRNA. And you can see it is expressed but barely. And when we test the firefly luciferase 2, which is improved from the wild-type, probably by around 100-fold. And from the firefly luciferase 2 mRNA, we optimize its codon and generate so-called high expressed firefly luciferase. And its expression at 24 hours post-transfection is about another 10-fold increased compared to the firefly luciferase 2, which means it is about 1,000 times increased compared to the wild-type firefly luciferase. In vivo data also shows the consistent trend that the high expression firefly luciferase is much better translated compared to the other two. We offer EGLP and the high expressed luciferase IVT mRNA as off-the-shelf product, and they can be purchased with small quantity and with or without the modified nucleotides. And they will be really good material to start your experiments if you haven't been used IVT mRNA before. And in your actual experiment, they could also be a good transfection control or positive control. Because they are off the shelf, they can be delivered in 2 to 3 weeks. Here's another good example shows the importance of codon optimization, that is comparing the COVID-19 spike gene versus BioNTech's spike coding mRNA vaccine. You can see that the red line indicates the amino acid, the two mRNA codes, and they are almost identical, but there are only 2 point mutation that the vaccine has, which helps to stabilize the configuration of the spike protein. And but when we look at the coding sequence, which is the blue background lines, you can see there are huge differences. And those differences that are found in the spike coding mRNA vaccine are critical for the function of the vaccine. So to produce the mRNA, not only we have to generate enough quantity, but we also need to make sure its quality is good enough for its expression. And then there are 3 critical checkpoints for its quality. The first step is to check the IVT mRNA vector. So once the cloning is done, you want to do necessary DNA sequence and RE restriction enzyme digestion to make sure the cloning is correct. The next step is after in vitro transcription, you want to do QC to make sure the RNA length, its sequence, and the capping efficiency are good. And also you can also test its translation efficiency, concentration, and purity. For in vivo gene mRNA delivery, the mRNA needs to be encapsulated in lipid nanoparticle. And if that is done, then you also have to apply necessary QC for the lipid nanoparticle. For example, you need to test the capsulation efficiency to make sure there are sufficient amount of mRNA encapsulated in the particles. Then you also need to test the size and the charge of the nanoparticles. So here are a list of detailed different choices of assays you can use for those QC tests. And we also offer them for research grade or preclinical grade mRNA. Here are some examples. This is a lipid nanoparticles under next standing TEM. And this is an example of doing dynamic light scattering to measure the diameter distribution of lipid nanoparticles. And this is an example of doing gel electrophoresis. You can see that the raw products, because this mRNA is really long, it's over 10 kb. So inevitably, you will see some truncated mRNA or degraded mRNA in the raw product. And after proper purification, you can see we collected really sharp, intact RNA bands. And this is another QC test on the integrity of poly-tail, because this is because the vector contains so many repeated poly-A, it is very unstable, and you'd have to do necessary QC to make sure it has enough amount of poly before in vitro transcription reaction. So we believe this is a platform of experiment-oriented development and optimization for mRNA drugs. You start with design your vector, which includes codon optimization, IVT vector, and the choice of 5' and 3' UTRs. After that is ready, you start with next step is to do synthesis and purification, including capping method, purification method, the choice of a modified nucleoside, and lipid nanoparticle packaging. And you also have to do necessary QC controls for the yield, for integrity, the double-strand DNA and double-strand RNA and the DNA residue quantification, the lipid nanoparticle profiling. And then you will take the product for experiments, including but not limited to in vitro and in vivo expression, mRNA stability, and the immunogenicity. And the information and the results you collect from quality control and experiments are really critical for you to further optimize your mRNA design and function. So Vector Builder provides a solution for IVT mRNA vector design and production. We can customize the production and the QC methods to meet the R&D requests from benchtop research to clinical trials. With the limited time, we can not only share more, but here are some more exciting RNA application. For example, in this case, we generate, instead of generating mRNA, we generated guide RNA using in vitro transcription, and we show that it can properly digest, it can properly combine with the Cas9 nuclease and cut the targeted position. And in this case, we use instead of mRNA, but a so-called self-amplifying RNA. This kind of RNA can replicate a little bit within the cell, which means the needed amount to achieve similar expression is even further less. And you can see its expression of this GFP is much higher than the conventional mRNA. Feel free to chat with us at booth number 15 if you want to learn more exciting technology about in vitro transcribed mRNA, or contact us at service-jb at vectorbuilder.com. Thanks for your attention. Please let me know if you have any questions.
Thank you, Charles. Any questions? Or you can stop by the booth number 15. So he's always there. So. それでは本日お集まりいただきましてどうもありがとうございます。